Hi, everyone. Thank you so much for joining SNEF Talk. Today we have Dr. Sara Cavallaro. We're delighted to invite her to give us a talk about Tyramide signal amplification strategy for fluorescent staining of single extracellular vesicles. Dr. Sara, thank you so much for accepting our invitation. So let me introduce Dr. Sara Cavallaro. Dr. Sara obtained her PhD in applied physics from KTH Royal Institute of Technology in Sweden. Her PhD focused on the development of different techniques for detection and characterization of extracellular vesicles at both bulk and single particle levels. Since February 2022, she's a postdoctoral fellow at Massachusetts General Hospital in Boston, and her work focuses on developing microfluidics and microscopy technologies for the detection and characterization of cancer cells and extracellular vesicles. She would like to use these advancements to better understand cancer mechanisms and to optimize patient therapies. So I'm Maimonah Al-Masawa. I'll be moderating this talk along with my colleague Débora Ferreira. We're very happy that you are joining this session. So, Sara, Dr. Sara, the floor is yours. You may start your presentation.
Okay, thank you so much, Maimona, for the introduction. Good morning, everyone. Good evening for some of you. As Maimona said, I'm Sara Cavallaro, and today I will talk about one of the works I developed during my postdoc in Shannon Stott lab at Mass General Hospital in Boston. So as Maimona already said, this talk is about the development of a signal amplification strategy for the fluorescent study and like staining of single extracellular vesicles. So I will not introduce what are EVs because all of you know about them. I want to just give a brief introduction about the topic by just saying that, as you know, EVs have a lot of cargoes inside that include like protein, nucleic acids, lipids, and thanks to these cargoes that reflect their parent cells and the fact that you can find EVs in body fluids, EVs represent a very promising source of liquid biopsies. Now, liquid biopsies in general are very valuable ways for disease diagnosis and monitoring because they are minimally invasive as compared to tissue-based biopsies. This is like true for all type of disease, but it is especially true for brain diseases because like it is very challenging to access the brain due to the presence of the blood-brain barrier. And so if you have a way to monitor the disease from like an external part, in this case, like from a liquid biopsy, instead of accessing the brain and do a tissue-based biopsy, this would be way more convenient. So in like my lab focuses a lot on brain cancers, specifically on glioblastoma. So this will be the focus of my talk. Glioblastoma is a very aggressive form of adult brain cancer that you can see, and as you can see here, it has a very poor prognosis. Like the 5-year survival rate is less than 5%. So, if we consider that EVs can cross the blood-brain barrier and like you can find them in circulation, and they can do it better than, for example, circulating tumor cells, because these cells can also be found in circulation, but it's more difficult for them to cross the damaged blood barrier. Then I hope I convince you that EVs can be a very promising source of liquid biopsies for brain cancer monitoring. Now, the question is, can we resolve EV heterogeneity and identify subpopulations? In order to do so, which is very important to understand EV function, and for example, if you're studying a certain type of disease, in my case, brain cancer, you would like to identify the EVs that are coming from the brain and from the cancer in the brain. You cannot use a bulk method, but you need to use a single particle technique. And the reason is that if you use bulk methods, you can have information about the average property of the sample, but you are not able to basically fully study one specific subpopulation of your sample. So for this, you need to do single particle techniques that allow you to study single vesicles. So here I listed a few methods that are currently used to analyze single vesicles, and they include digital PCR. You can have interferometry-based method and microscopy-based method. For example, like flow cytometry, immunofluorescence staining, and scattering. In my work, I focused on immunofluorescence staining because we believe this is a widely accessible and convenient method that basically all the labs have. All the labs have a fluorescent microscope, and most of the labs also have a confocal microscope. So we wanted to develop a method that is widely accessible to people. And so that's why my talk focuses on an immunofluorescence staining method. So if we focus on this, of course, the major challenge is that if we look at the EVs, they are very small compared to cells. So it means that they have a very limited number of cargoes that are carried by the vesicles. So the challenge is that the fluorescent signals are usually very weak. And as you can see in this image, they are very unstable over time. So it means that if you are trying to stain your vesicles with this method, you usually don't even have the time to look at your sample that the signals have already disappeared. So there is a need for a technique or like for techniques that are able to amplify and stabilize your signals. So that's the focus of my work. So the aim of my study was to develop an immunofluorescence staining technique, which in my case it's called the TSA because it's based on a Tyramide signal amplification that was able to amplify and stabilize the fluorescent signals generated by single vesicles. So this work was just accepted in the Journal of EVs. So if you want to have further information, you can also read like this online. So here you will see a schematic of like before I go into like the details of my TSA method, I will introduce other conventional immunofluorescence staining methods that are used to study single EVs. So these are two methods that are used like in the literature, and they are called the direct staining or primary secondary staining. So the way this method works, in case of direct staining, you have your protein of interest and you just use an antibody that has a fluorophore attached to target your protein or like your target of interest. In case of the primary secondary staining method, you have a primary antibody which is unlabeled, and this like is followed by a secondary antibody that binds to the primary antibody, and it's this secondary antibody that has a fluorophore attached. So the way the signal is generated is by the fluorophore carried by these antibodies, and usually, I mean, depending on the antibody you buy, you can have one fluorophore or like a couple of fluorophores attached to an antibody. But these are the only, like for each target, this is the only fluorescent signal that you will get. So my method, which again is called TSA because it's based on these Tyramide probes, is you can see a schematic here. So the way it works is that you have a primary antibody that is unlabeled. Then you have a secondary antibody that is also doesn't have a fluorophore but has a HRP. And like the way it works is that you have small probes that are the fluorescent probes that are called the TSA probes. These get activated when they are in proximity of the HRP, of the secondary antibody, and they deposit very close to your target protein. So the way the signal is, if we now like look at these more into details, the way the signal is amplified is that because for each pair of primary secondary antibody, you can have multiple Tyramide probes that get activated and deposit in proximity of your target. And so it means that you have multiple fluorophores for each protein. And that's how the signal gets amplified. Here you can see two colors because these probes are available. First of all, they are commercially available and they are available in different colors. So they give you the possibility to multiplex the techniques because you can have like one protein targeted with one color and then you can use another color to target a different protein on the surface of the EVs. So this is the way our method works. And then of course, in addition to developing the protocol, the staining protocol, we also had to work on the development of the immobilization protocol that allowed you to have the EV stably immobilized on the on your substrate. So here you can see a schematic of the method. So you can see like there is a cell. So in my study, it was like a glioblastoma cell line to start with, and then we move to patient samples. You isolate the EVs using in our case, we used a combination of filtration to concentrate also the sample and the size exclusion chromatography through the ISON column. And then we developed a protocol to immobilize the EVs on the surface. It was just, I will show you some further details later, but it's basically just an absorption on a glass slide using overnight incubation. And once the vesicles were stably immobilized, you could proceed with the staining method and then with the imaging. So to start with, before applying our method on the EVs, we wanted to validate it on cells. So what you can see here is that we started with our glioblastoma cell line. We isolated we concentrated then basically isolated the cells using some centrifugation, and we mobilized them on a glass slide. And what we wanted to do first is we wanted to see if our developed PSA method was providing the amplification of the signal compared to the other two methods that are the direct staining method and the primary secondary method. So for this study, we stained the cells. Like, first of all, we used 3 different substrates where we immobilized the same number of cells on them. And then we used DAPI to stain the cells. And then we wanted to test our method versus the other one by using a marker that we know is expressed on the surface of the cells, which in our case is EGFR. So we had validated with other methods that these cells express EGFR. And what we did was to basically stained target EGFR using our method, the TSA method, and then using the other two methods, direct staining and primary secondary staining. And for this, I would like to highlight that basically we use exactly the same conditions, like the same concentration of antibodies, the same type of antibody, and like the only difference was that like the TSA had the probes, but like the clones of the antibodies were all the same in order to reduce like changes or variability due to the different like reagents. So as you can see here already, like visually, you can see that like the TSA method provides like in the TSA method, the EGFR signal, which is the green signal around these cells that are pointed by the green arrows, is way stronger than in the other two cases, like direct staining and primary secondary staining, where you can weakly see some green signals, but they are weaker than like the TSA method. So you can already see visually that the signals seem to be amplified. When trying to quantify these signals, what we did was to basically calculate the integrated fluorescent intensities on single cells, and then basically plot these for all the cells that were available in our slides for the 3 techniques. And what you can see on the plot, in the plot on the right, is that the TSA method provides more than 13 times amplified fluorescent signal intensities for EGFR as compared to the primary secondary staining method. Of course, it was even more for the direct method only, which is the weakest. And what was interesting to see was also that the dynamic ranges were also amplified more than 25 times. And this is very important because sometimes when you develop an amplification method, you have an amplification of the fluorescent signals, but you don't amplify the dynamic ranges, like they remain the same. In our case, also the dynamic ranges were amplified, and this is important because it means that you can better study variations in the signal expression between different samples. So this gives you more sensitivity in your technique. After this, we wanted to see how stable are the signals generated by the TSA method compared to the other two methods. So what we did was, by using like a confocal microscope, we excited these cells for like 1 minute, 3 minutes, and 5 minutes, like of continuous excitation, and we like calculated basically the histogram and like the distribution of the fluorescent signals over these different times. And what you can see in the plot on the bottom part is that in case of cells, first of all, the TSA method was very stable. You can see that like even after 5 minutes of continuous excitation of the sample, the signals were still there and they didn't decrease over time. But in case of cells, also with the other two methods, the signals were really stable. And the reason is that cells usually have like a lot of markers on the surface, so you don't really have the problem of the instability of the signals. But this will be very important when we move to EVs. So after validating that like the signals were amplified with our method, we wanted to see if it was possible to have some multiplexing on the cells. So for this study, we did two different experiments. The first experiment was to see if we could use the method on like to use two different Tyramide colors, Tyramide probes, which was a green probe and a red probe to target two different type of cells on the same substrate. And what you can see on the top part of the slide is that we basically took our glioblastoma cells, we mix them with white blood cells from a healthy donor. We plated these two together on a slide, and then we used these two different Tyramide colors to target these two different cells. Now, for the GBM cells, what we did was we used a combination of, instead of using just one primary antibody, we use a combination of 5 primary antibody because this is a cocktail that we have seen in our lab, like we have developed in our lab as a specific cocktail to target glioblastoma cells. So the reason why we use this is because we know that this cocktail allows you to identify glioblastoma cells in a complex patient sample. And it was targeted using the green probe. And then we used the CD45 and CD11C to target with a red Tyramide probe to target the white blood cells. And as you can see, like on these two images, the signals were really specific. So the green targets, the green probes were only targeting the bigger glioblastoma cells, while the red probes were targeting the smaller round white blood cells. So these results seem to show that it was possible to use like this protocol could be multiplexed. We also did an additional experiment because then like this was useful later when we brought our technique on the EVs. That was like, can we use like these two different colors to target two different markers on the same cell? So we used again the team cocktail to target in green to target like the more specific glioblastoma markers on the cells. And then we use also for the red probe, we use a combination of CD9, CD81. And we wanted to see, can we see on the cells and like, can we basically use this duplex color on the same substrate? And the results seem to show positive outcomes because as you can see, like it was possible to see these two colors on the cells. Of course, the transferrins were not super expressed because this again also depends on the sample, on the tetraspinin you select, but the results seem to be promising. So once we validated the protocol on cells, we were ready to move to the EVs. And to do so, we first wanted to characterize our vesicles. As I said, we started with EVs that were isolated from a glioblastoma cell line, and we isolated them using ISON. So following this isolation, we performed the general EV characterization method. So we did NTA, which showed the presence of like EV-sized particles, and then we did ELISA and also DSOR imaging through the ONI system to look at like more specific EV markers. So we look like at the tetraspinins, CD9, CD81, CD63, and we also looked at EGFR, which again, we know that is expressed by these vesicles. And what you can see here is that there seem to be EVs in our sample because, like, as you can see, for example, in this image on the right, in the ONI image, there seem to be particles that express the tetratraspanin with different expressions, but these results confirm the presence of EVs. Following this, we had to optimize our immobilization strategy because compared to cells where it's kind of known how to immobilize cells on a substrate, in our case we use cytosine. With EVs, you also need to really optimize your immobilization protocol. And what we did was we used some fluorescently labeled vesicles. They had the CD63 tagged with a TD tomato fluorophore, and we use these vesicles and we plated them on different type of substrate, they had different coatings and also different concentration. And what you can see here, this is the result for our best substrate. So the best substrate was basically a glass slide that is commercially available. It's called Trubon. It has an adhesive layer, and by basically plating these vesicles on the slide and leaving them just overnight, they were absorbed on the slide. They were stably basically immobilized. What you can see here is that there are like, when we looked at these vesicles, there were like these dots that corresponded to the vesicles. We could see that this slide was retaining a good amount of vesicles, first of all. It was also showing the effect of different concentrations that we were expecting to see. Because like we tried different concentrations to see if we could detect like different number of vesicles on the two different substrates, and we could see this. We also could see that like the PBS control, that was a control like slide without any vesicle, didn't show any result. And what was important is also that this slide was very good in retaining the vesicles over multiple washing steps. Because when you do a staining protocol, usually you need to, you have like multiple steps of like staining, then you need to wash out your antibody, then you need to wash like with PBS. So it's important that your substrate keeps the vesicles attached for as long as possible. So then you don't lose your vesicles while doing the staining protocol. So this is the slide that gave us the best results. So we moved forward with this type of substrate for all the subsequent experiment. So what we did, as in case of cells, so with EVs, we wanted to see how the TSA method compares with the other two conventional methods of direct staining and primary-secondary staining. So we first isolated the vesicles using ISON columns, and then we plated on three different substrates that were these adhesive slides that I mentioned before. And we stained these vesicles targeting CD63 because we knew that these vesicles express CD63. We stained them using the three different methods. And what you can see on this image is that, again, as in case of cells, the TSA method provided way more intense signals compared to the other two methods. And for EVs, I think these results were very interesting because they tell you that, like, they highlight you the importance of the technique when you want to stain your vesicles, because if you were using like a direct staining approach, so you would only, like you were trying to characterize your vesicles by using a conventional technique, you would barely see any dot on your substrate. So the first thing you could think was that maybe your vesicles are not expressing that marker because you are not seeing any fluorescent dot. But this doesn't actually, what we saw is that this doesn't mean that your vesicles don't express the marker, it's just that your method is not sensitive enough to detect those markers. And so you need to often use an amplification method to actually really see the expression of the marker on the surface. And this is what we were seeing with the TSA method. You see, you have way more dots corresponding to single vesicles as compared to the other two methods. And in all cases, the PBS control substrates were very clean. So this meant that the signals were specific. Following this visual inspection, we also wanted, so in case of like EVs, what you can see here is that the signals were amplified more than 6 times. Of course, the amplification factor with the TSA method compared to the other two methods. Of course, the amplification factor is lower compared to cells, but this is normal because, like, of course, the EV surface is way smaller than a cell surface, so you will have like a lower number of markers available compared to the cells. We then quantified the fluorescent signals on single vesicles and plotted them for all the vesicles that were available in the like in those fields of view. And the results showed that the TSA method show 3 times amplification of the dynamic ranges as compared to the primary secondary staining method. So even in case of EVs, the signals were amplified and the dynamic ranges were amplified, so it was possible to detect a better variation of the signals among different samples. And when looking at the stability of the signals, what we saw is that with the TSA method, the signals were very stable over time. That is what you can see here on this plot, bottom right part. And especially this was way better than like the direct staining method, where you can see that the signals were very unstable over time. So they were already weak. And by the time you were trying to image your samples, they were really disappearing. So as I said before, in case of EVs, the TSA method gave was more advantageous because it was allowing you to have the signals very stable over time, and you could image your sample without affecting your results by the fact that the signals were decreasing and disappearing over time. So once we did this first verification with a commonly expressed EV marker, which was CD63, we wanted to see, okay, can we use now our method to detect low abundant markers on the vesicles? And for this, what we did was we took EVs that were isolated from 2 different cell lines, glioblastoma cell lines, which here I just called GBM1, GBM2, and we used the TSA method to target 2 different low abundant markers expressed by these cells, which is MET and EGFR. So what you can see here is that the method was able to detect these markers on the surface of the vesicles, and also it was able to detect differences in the expression of these markers on the EVs. This was actually one of the reviewers' question about like this work, because they were asking like, okay, can you actually detect differences in 2 different samples when you have low abundance marker? And as you can see in this plot, there were significant differences between these 2 samples for both markers. So it seems that the method is working very well on the EVs. After this, we wanted to see can we actually multiplex this method on the vesicles? And to do this, we did a preliminary experiment where we wanted to try the 2 different TSA colors on 2 different substrates. So we plated our vesicles on like 2 substrates, and we stained one substrate for the steam cocktail, which again is this cocktail of 5 antibodies that we use in our lab to detect glioblastoma specific markers. And if you want to have further information, you can read this about the paper but it's basically a combination of 5 antibodies that are like SOX, tubulin, EGFR, A2B5, and MET. So we call this like the GBM specific marker. And then we did another substrate where we put CD9 and CD81 targeted in red to see the EVs. And these preliminary results were positive because it showed a positive expression of this marker of both colors on the substrate with EVs as compared to the control substrate. This was important because when you have an amplification method, sometimes you have also an amplification of your background, and especially when you do then a cocktail of antibodies, the unspecific staining sometimes really increases. But in our case, that the signals seem to be very specific, even in presence of like a cocktail of antibodies, which were like in an amplification strategy method. So the signals seem to be very specific and also quite stable over time. So once we tested these 2 different colors separately, we wanted to say, okay, now can we actually multiplex these on the same substrate? And for this, We prepared a substrate with EVs and we stain it using first the green probe for the steam cocktail of markers, and then this was followed by the red color targeting CD9 and CD81. So what you can see here on the top part of the image is that we could see like single EVs on the substrate, and there were some that were only expressing the tetraspanin, some that were only expressing like the steam markers, and some that were co-expressing both. When we tried to look at like to detect the number of EVs for two different concentrations, the results seem to be following our expectation because we could detect a variation in the number of EVs for these two different concentrations, which means that you are mostly targeting single vesicles. This is one of the checks that you usually need to do when you study single EVs. You try different concentrations and you see if your signals are following, like the numbers are following the expectations, because it means that you are looking at single EVs. But what we realized in this case was that when using this two-plex staining protocol with the vesicles, we had some unspecificity. And the reason was that, as I said, we started with the green color. So we basically targeted the GBM specific markers, and we put the green PSA probe. And then when we were putting the red probe that was targeting the CD9, CD81 markers, we saw this is what you can see here in this first plot. We saw that there was like 20% of the signals that were coming from the red probe were actually unspecific signals. And these were generated by the fact that the second Tyramide probe was reacting with some residual active HRP, which was present on the first staining cycle. So basically, these signals were not really generated by the tetraspanin, but were generated by some cross reaction between the first pair of antibody targeting the STEAM marker and the second fluorescent probe. So with this experiment, we understood that we had to further optimize our protocol in case of EVs when doing multiplex staining, and we found out that by using like a quenching buffer, that it was like a buffer based on hydrogen peroxide at a specific concentration and for a certain amount of time. By using this, it was possible to reduce the unspecific signals to non-significant levels. This is what you can see on the plot, like that I am pointing with the arrow here. So basically, by using this quenching buffer, it was possible to remove this a specific staining that was coming from the second staining cycle. And also the signals were not affected. So what we wanted to check is this buffer also kind of decreasing our signals? And the results were like negative. No, this buffer was really good in like removing the unspecific staining, but still keeping the same signals that were detected without the use of this buffer. So we had to introduce, in case of EVs, this quenching buffer to make our multiplex protocol working properly. So after we validated these on the EVs detected, like isolated from a cell line, we wanted to move to patient samples. And our question was, can we use now our developed method to stain EVs that are coming from glioblastoma patient samples? And for this, what we did was we had access to plasma samples of glioblastoma patients. We isolated the vesicles using ISON columns and the filtration. And we did the characterization methods of like NTA, TEM, and Western blot. So as you know, when working with patient samples, the situation is way more complex because you can have a lot of like contamination by the lipoproteins. So you need to do your best first to decrease this contamination. So our preliminary characterization results show that with our isolation method, we were detecting particles in the EV size range, and we were also detecting particles that had the double lipid layer. So basically, we could see that we had EVs. We also saw that some patients had like some lipoprotein contamination. It's not super clear on this image because it's small, but basically we were seeing like some lipoprotein contamination in these patients. The amount of contamination depended on the patients. There were some with very little contamination, some with higher contamination. But what was nice to see by Western blot was that in all the cases, like the lipoprotein contamination really decreased in the EV preparation as compared to the plasma sample that we had at the beginning. This is what you can see here on the Western blot when targeting the ApoA1 lipoprotein. We saw that the plasma sample had a lot, while the EV sample had way less than the original sample. So we were successful in decreasing the lipoprotein contamination, even though there was still some on the EV sample. So after characterizing our samples, we applied our TSA method on the EVs isolated from these samples, and also we compared this with the CTCs that were isolated from the same plasma sample. So what you can see on the first part is that the TSA method was really good in detecting the circulating tumor cells. So these were basically cancer cells released by these glioblastoma patients, by basically in their blood, because you can see here like this green big cell is a glioblastoma circulating tumor cells, and it was very clear that it was like targeted in green, while the white blood cell again was targeted in red. So the cells, like the cells results showed that we could detect by using our TSA method, we could detect like circulating tumor cells in 59% of the patients that we analyzed, and compared to healthy donor, where you see like all the results show that like the healthy donor had zero count. So this 59% detection rate might seem like not the best, but is actually very good considering that circulating tumor cells are very rare in circulation. And it was also, I want to highlight that it was double the detection rate that we could detect in an earlier study where we were targeting and staining these cells using a conventional method of direct staining. So this again highlights the fact that with our TSA method, the signals are amplified and you can increase the sensitivity and the detection rate. When we looked at EVs, which is the lower part of the plot, first of all, I want to mention that we could only analyze 5 patients compared to all the patients for which we had plasma. So we analyzed 5 patients, and we use again the same cocktail to look at the glioblastoma derived EVs and the general EV marker CD9, CD81 to target EVs, and to see if we could detect GBM EVs in the plasma of these patients, we used a double positivity strategy. So we only considered positive the EVs that were double positive for both these colors. And the reason is that this would allow us to decrease the basically false positive that could come from the lipoprotein contamination. So after using this double positivity strategy and after also using some control substrate as basically comparison as our controls. So there were some healthy donors that didn't have any brain cancer. We could see that we could detect glioblastoma EVs in 2 of the patients. This is the last column of the table. So we could detect GBM EVs in 2 of the patients of the 5. And this was these were the same patients that showed the highest count of glioblastoma circulating tumor cells. So basically, the results seem to make sense and to match the results on cells. Here, I would like to mention that we didn't detect GBM EVs in the other 3 patients, I think because of a limitation of the imaging, not of the technique, because when you look at cells, you can basically image your whole slide with the cells because there are automated microscopes that can scan the whole slide, but this is not possible with EVs because you usually since the signals are so small, the microscopes can start acquiring the images automatically, but then after some time they lose the focus and they are not able to find the focus anymore. So they can't really scan the whole slide. So we could only basically acquire a limited number of field of views on these slides, and that's the reason why I think we couldn't detect GBM EVs in all the samples, but of course, this needs to be further optimized in the future. So to conclude, I hope I show you that the developed TSA method shows an amplification of the fluorescent signals. It shows broader signal dynamic ranges and also a better stability of the signals compared to conventional methods using either primary labeled antibodies or primary and secondary labeled antibodies. We showed the first multiplex staining of single EVs with this method because there were there were like two studies that were starting to use the TSA method on EVs, but they didn't show any multiplexing. And also we showed a successful staining of plasma samples using this developed method. Of course, in the future, we would like to further optimize the method for the analysis of patient samples. There are many things that can be optimized. We would like to increase our multiplexing capabilities, and we would also like to integrate our staining protocol with some more specific isolation of the vesicles. In our lab, we have developed a microfluidic device based on EV immunocapture to further isolate some population of vesicles. So we would really like to integrate this staining protocol into the microfluidic device so we can study even more specific subpopulations. And of course, we would like to expand the cohort of markers and diseases that can be analyzed. So with this, I would like to thank my research group, my PI, Professor Shannon Stott, my whole research team at MGH. I would like to also thank our collaborators at MGH and also the Mayo Clinic. I would like to thank the patients who gave consent to use their samples. And I would like to thank the funding agencies, especially the American Cancer Society, for providing me with a postdoctoral fellowship to support and perform this work. And last but not least, I would like to thank you all for your attention, and I will be happy to answer any questions. Thank you.
Thank you, Dr. Sara. Fantastic talk. It was really an insightful presentation, very comprehensive and educational. The session is open now for questions. You can either unmute yourself or put your questions in the chat box. I would go first. So my question is most related if you did you observe differences in amplification efficiency depending on the EV size or the biogenesis pathway? Have you tested?
So we, for the moment, didn't test different biogenesis pathways because we were just basically isolating the EVs with ISON, and usually it gives you everything that is released in this size range. But we could see that, yes, the amplification factor depends also on the markers that you are analyzing because I think this is also dependent on the antibodies, the primary antibodies have different affinities. So I think also depending on the affinity, let's say if that affinity is not the best, you might have a lower amplification factor for that marker compared to another one. So yeah, it's dependent on that. We didn't have time to try so many markers for example, or so many antibodies for the same marker, also for a cost limitation because antibodies are always super expensive. But I believe, yes, it's dependent. But that's why we wanted to let's say prove that our method is providing an we wanted to basically show mostly that if we keep the same antibodies, so you select a certain gene, a certain clone, a certain antibody that is targeting one marker, if you keep the same and then you use the three methods, you have an amplification of the method. But yeah, the efficiency then depends on the marker.
Thank you. We have here a question. I think Vietnam want to ask the question. You can unmute yourself if you want.
Thank you so much for the nice talk. I have a couple of questions. My first question is, what type of EVs were you targeted, in the size, the larger or the smaller ones? Because I would like to know this multiplex assay that you developed, for how many markers at a time was it usable, including the tetraspanning markers? So these are my questions. Thank you so much.
Yeah, so for the size, thank you so much for the question. I will try to go back. Let's see. Okay, yes. So for the size, we were mostly having an enrichment of small EVs this is what you can see both with the patient samples here. Like you can see that most of your vesicles are like less than 200 nanometer. And also like I'm trying to go back to the cell lines. So this is like the results from the cell lines where again you see like a enrichment of the small EVs. We also had some larger EVs, but with the ISON usually you reach like considering the fractions that we were collecting, we would see EVs up to 1 micron. So I would say that my upper size limit was 1 micron, and the lower size depends on the sample. And I think we had like 150 nanometer of EVs. And I think this is also where the technique is mostly valuable because, I mean, since you have a very small surface on these EVs, you will likely have like less markers available. So it's where like the amplification of your signal is most valuable. And the other question like was about the markers. So for the moment we used like two different so you basically can target, let's say, you can have different strategies because I think like the multiplexing capabilities depends on the how many colors you use on the same substrate. We only tested two. I think like we would like to test more, and I think you can test like three or four, but I don't think you can really increase this too much because of the fact that the EVs are very small, so compared to cells. So since this method requires a deposit, like you have the deposition of the probe in proximity of the surface, I don't think you can overcrowd like the surface of EVs with so many different colors. So the number of colors depends on your I think on your needs, and like it can't be more than like three or four. And the markers see depends like because as what I showed is that we could use one color to just test one marker in case of CD63 or in case of EGFR MET, but you could also test one color for a cocktail of like here you have one marker only with one color, or you can have like a combination of markers for a single color. So this depends like on your needs.
We also have here some questions on the chat box. We have one from Bhuvan that asks, how do you ensure that the TSA signal reflects the true glioblastoma specific expression and not amplification artifacts?
Yeah, so I think this you can do in many things. That is like all the controls we did. Like if you have just amplification artifacts, like you will see that in your both control surface where you have just substrates without the vesicles, and you stain these using your same like antibodies and like probes, but especially you can like I think the real control is the healthy donors. So what we did was we also tested some healthy donors that they had EVs, and we used the staining protocol on these healthy donors. And then what we did was we were seeing a few positive signals that we were using like as a threshold to detect positive instead, like let's say above this value is positive for the GBM samples, below this value is negative because it's some again amplification artifacts or like some just unspecific signals that you can have also in like healthy donors. So I think it's like by doing multiple different like controls you can ensure that your signals are most likely specific.
And then you have another one from Bhuvan asking that glioblastoma cells are highly proliferative. How does this could affect the TSA staining?
Yeah, I'm not sure like about like how this could interfere because I mean like yeah the even if they are highly proliferative is like I mean we are targeting some markers on their surface, so I'm not sure about this question like about how the TSA interfere. I don't know if Bhuvan wants to if he has more details on the question.
I don't know if it is already here.
Hello.
Oh, yeah.
Yep.
Yeah. So when the glioblastoma cells are highly proliferative, so then it increases the tyrosine residues and metabolic enzyme, so that it leads to the non-specific pyrimidine deposition. So how you tackle with this?
Uh, I mean, I think that okay, even if you have more sites to bind, the reaction is still active. So the reaction is not activated by what is on the surface of the cells, but by the fact that you have a primary antibody that is binding to your specific to your protein of interest. Let's say you have EGFR, so you use the primary antibody to target EGFR. Then you have a secondary antibody that binds to the primary antibody, so it's not binding to like the surface of the cells. And then once you have that, the secondary antibody activates the probe. So that's why like you can have like the binding itself depends on like the presence of these antibodies. And of course, we did a lot of controls where we also tried to see, okay, what if we just put the secondary antibody without the primary antibody? Will this create signal unspecific? And we didn't see any unspecific signals, even in case of these GBM cells that were mostly the cells I used for my studies. So I think it's because you have these chain of antibodies that is generating and activating the signals that you can also stain highly proliferative cells.
Thank you, everyone. We also have another question here at the chat from Josephine asking, have you tried using the method on EVs just isolated from tissue? For example, EV isolated from tumors?
So no, we haven't tried these on EVs, like from, if you mean like in tissue sections, no, this would be like our next also work, like could be one of our next works, because we have this technique which is slightly like it has some adjustments validated with cells like on the tissue sections. It's slightly different, but it's very similar like approach. So we can stain with this technique like cells on the tissues, but we haven't tried with EVs. But maybe I was really interested in seeing if we could do it. So I think in the future we will explore this possibility.
We have another one from Anais. He's saying, thanks a lot for this very interesting talk. I'm not familiar with this steam cocktail and which markers it targets, but is it possible to target also intraluminal markers with ESA?
Yes, you can do it because you have some like a slightly permeabilization that you can do, and you can do these in our buffers. So actually we have done this with like cells and also with EVs. You have some slightly permeabilization that allow you to go to basically the antibody to go inside. With cells, it works really well. With EVs, we still have to check like I think for single markers, because I don't know also like how would the antibody going inside the vesicle work like, but in principle it's possible because we have some like slightly permeabilization for this staining protocol, and basically these buffers are also like they are not destroying the cells or the EVs, but they are just slightly permeabilizing them. And so for the other question that is like combined with other method.
Alsana also have another question saying, would this amplification technique could be combined with other single EV microscopic approaches, such as the D-storm with the blinkable fluorophores?
Yeah, no, I don't think this one is can be used in the D-storm, because again you need the blinking behavior, and it has to be also like some specific blinkings, like not even like in the D-storm images you cannot use any type of fluorophores, so you will need some specific blinkage behaviors and some specific blinkage buffers. So at the moment, no, we cannot use this with the D-storm technique.
Thank you, Sara. I also have here another question from my side, just to see your opinion. How robust do you think that this approach could be to variability in different EV preparations from different biological sources?
So a different EV like biological sources in terms of like human or like CSF versus
Yeah,
plasma. Like
urine, for instance.
so we have only tested plasma, and I've started some investigation with CSF, and it seems to be working. I haven't tried like urine, so I think let's say for different biological preparation, we might need to further optimize the blocking part, because I
Yeah.
think some different biological samples might have more like contamination, less contamination, or different types of contamination. And we might need to adjust the blocking buffer, but theoretically, I think the technique would work. It's mostly like trying to tune the blocking buffer so then the specific staining is not too high, or like it's decreased as much as possible.
Okay, that's great. We are just a long time. I would like to thank everyone that for joining this session and for the great questions and discussion. A special thanks, of course, to Dr. Sara for her outstanding presentation and also for sharing these valuable insights. It was truly inspiring. You can then see this recorded session that will be available soon at YouTube. I would like to say that have a nice week, all of you. Okay, and thank you, Dr.
Thank you
Sara,
so
once
much.
again.
Thank you so much for the invitation, and thank you all for being here. Thank you.